Hello everyone, and today we are starting with the new session of the topic photosynthesis in higher plants, and we'll be studying about light reaction in detail today. So let us first discuss about the structure of the chloroplast. So chloroplast is basically a double membrane structure. It consists of an outer membrane and an inner membrane. Okay, and thylakoid consists of grana. This entire thing, this is grana, which are made up of thylakoids. Okay, and this individual one is thylakoid. These are thylakoids which are stacked over each other and they form the grana. So thylakoids are nothing but they are disc-like structures which are kept over each other, or you can say stacked over each other to form a grana. These thylakoids are the site of light reaction. Okay, light reaction takes place here. Now, what is stroma? Stroma is the colorless fluid present inside the chloroplast. This is known as stroma. And we can also see some ribosomes, starch granules, and some lipid droplets in chloroplast. But the point here to remember is that chloroplast consists of thylakoid which are stacked over each other forming a grana. Thylakoids are the site of light reaction. And then we have this stroma lamella. Okay, so these stroma lamella, these connect the two thylakoids. Okay, this is thylakoid, this is another thylakoid. So these stroma lamella connects the two thylakoids. We'll be studying about the stroma lamella in later, and we will discuss about the importance of stroma lamella. Now photosynthesis. Let's talk about photosynthesis. Photosynthesis is completed in two phases. First one is the light reaction. Second one is the dark reaction. Today we'll be discussing about the light reaction. So as I've already told you, the site of light reaction is thylakoid disc, you can say, or you can say the grana of the chloroplast. Grana is made up of stacks of thylakoids. So either we can say thylakoid disc. And why we are saying thylakoid disc? Because thylakoid are disc shaped. That's why thylakoid disc. Or we can say stacks of thylakoid, which is nothing but grana. And the dark reaction takes place in the stroma of the chloroplast. Stroma, I've already told you, is the fluid of the chloroplast, fluid present in the chloroplast. Now let us discuss about the mechanism of photosynthesis. Now again, let's discuss about the light reaction or light phase and the dark phase. Light phase is also known as the photochemical reactions. It is known as the light dependent phase or the reaction. It is also known as Hill's reaction. Whereas the dark phase comprise of the dark reaction, we can also say light independent reactions, Blackman reaction, and it is also known as biosynthetic phase because there is synthesis of the carbohydrates here. That's why it is known as biosynthetic phase. So what happens during light reaction? The energy stored in the sunlight is converted into chemical energy in the form of ATP and NADPH. Okay, so the energy stored in sunlight is converted into energy rich molecules which are ATP and NADPH. Whereas in dark reaction, which take place after light reaction, both ATP and NADPH which are produced during the light reaction are used to synthesize the sugars, or we can say carbohydrates. In light reaction, what happens? Oxygen evolution takes place in light reaction, whereas in dark reaction, carbon dioxide fixation, or you can say carbon dioxide reduction takes place. Okay, there is a reduction of carbon dioxide, leading to the synthesis of the sugars or carbohydrates. The main sugar that is produced is glucose. In light reaction, both photosystem 1 and photosystem 2 are involved. We have already discussed about photosystem 2 and 1 in earlier slides. And whereas no photosystem is involved in dark reaction. Whereas both PS1 and PS2 are involved in light reaction. We'll study about this PS1 and PS2 in later slides. So this is the general mechanism of photosynthesis. Here the light is captured by the pigments which are present in the thylakoid. So this individual one is the thylakoid, Okay, this forms the grana, and this thylakoid are forming the grana. Light is absorbed by the pigment present in the thylakoid membrane, and water is also involved here. Photolysis of water takes place here. ATP and NADPH are synthesized during light reaction, and this ATP and NADPH are used in the dark phase or the biosynthetic phase. So this one entire is the light reaction or the light phase, and from here we have this one. This is the dark phase. Okay, in dark phase what happens? The ATP and NADPH, which are synthesized during the light reaction, are used for carbon dioxide fixation, and this leads to the synthesis of mostly sugars, fatty acid and other amino acids. Now let us discuss a little bit about the electron transport chain, and we can electron transport chain we also call it as Z scheme. Okay, because it is like zigzag. So electron transport chain is nothing but it is a sequence of protein complexes and electron carriers, okay, which are situated within the membrane, and these protein complexes and electron carriers, they facilitate the transfer of electron in a controlled manner. Okay, these electron transport happens in a zigzag manner. That's why it is known as Z scheme or Z scheme. Okay, so what happens? The electrons are transferred. This is a very important point to remember. The transfer of electrons takes place from a molecule with low redox or reduction potential to a molecule with high redox potential or reduction potential. Okay, so what is reduction potential? Ability to gain electrons or ability to get reduced. Okay, so gaining of electron is known as reduction. So electrons are transferred or transported from a molecule with low reduction potential to a molecule with high reduction potential. So next term is phosphorylation. Phosphorylation is just the synthesis of ATP using ADP and inorganic phosphate. Okay, inorganic phosphate in the presence of light. So phosphorylation is generally of two types. First one is cyclic photophosphorylation and second is non-cyclic photophosphorylation. So now let's discuss first about the cyclic photophosphorylation. So let's study about some of the basic points in cyclic photophosphorylation. So in cyclic photosynthesis, what happens? Only photosystem 1 is involved. This is the point to remember. Only photosystem 1 is involved, which consists of lysed harvesting complex 1. Okay, this photosystem 1 is only present in the stroma lamella. Okay, that's why we can say cyclic photophosphorylation occurs in the stroma lamella. And what was stroma lamella? Stroma lamella was nothing but it connects the two thylakoids. Okay, it is present in the chloroplast. The electron ejected from the reaction center, which is again chlorophyll A. Okay, which show maximum absorption at 700 nanometer. Okay, so the electron ejected from the reaction center of photosystem 1 returns back to the reaction center. That's why it is known as cyclic photophosphorylation. Okay, I'll show you the diagram in just later slides. And another point to remember here is that no oxygen evolution takes place during cyclic photophosphorylation because there is no splitting of water. The water splitting complex is only present in photosystem 2. Okay, that's why there is no oxygen evolution in cyclic photophosphorylation. And another point to remember is that only ATP is generated here and no NADPH is generated in cyclic photophosphorylation. And the primary electron acceptor, you can say the first electron acceptor, now we know is the ferredoxin reducing substance, FRS. Okay, later it was thought, earlier it was thought that ferredoxin was the electron acceptor, but now we know that primary electron acceptor is ferredoxin reducing substance or FRS, which is nothing but an iron sulfur containing protein. Okay, so this is a flowchart of cyclic photophosphorylation. So what happens? Only photosystem 1 is involved here, which has the reaction center, which consists of chlorophyll A, which has the maximum absorption at 700 nanometer. That's why we can write this as P700. Photosystem 1 is present in the thylakoid membrane. It absorbs the sunlight, okay, absorbs the energy from the sunlight, and it gets excited. The chlorophyll A, which is the reaction center, get excited. It releases electrons, which is accepted by the primary acceptor, which is made up of an iron sulfur protein. Okay, so this is the primary acceptor or the first acceptor. Primary acceptor of electron. It is an iron sulfur protein. It is an iron sulfur protein, also known as FRS or ferredoxin reducing substance. Okay, and then we have ferredoxin. Earlier it was thought that ferredoxin is the primary acceptor, but later it was found that ferredoxin reducing substance is the primary acceptor. So what happens in cyclic photophosphorylation? The electrons which are ejected from the photosystem 1, okay, it is returning back. It is following this pathway and it is returning back to photosystem 1 here again. And during the process, only ATP is synthesized and no NADPH is synthesized. Okay, so here electrons are transferred to ferredoxin from FRS. Then from ferredoxin, electrons are transferred through to cytochrome B6F complex. Okay, and from this complex, it gets transferred to plastocyanin. And from plastocyanin, again, the electron return back to the photosystem 1. That's why it is known as cyclic photophosphorylation. The electron released from photosystem 1 return back to the photosystem 1. Now let us talk about the non-cyclic photophosphorylation, which is again called the Z scheme. Okay, because it follows the zigzag pattern or it makes a Z pattern. Here both photosystem 1 and photosystem 2 are involved. Okay, and the site of occurrence of non-cyclic photophosphorylation is grana, thylakoids. Okay, or you can simply say grana. The electron ejected from the photosystem 2 never goes back to photosystem 2. Okay, so like in cyclic phosphorylation, electrons ejected from PS1, photosystem 1, return back to photosystem 1. But here, what happens? Electrons ejected from photosystem 2 don't return back to photosystem 2, but it is accepted by the NADPH. Okay, and NADP plus, it is accepted by NADP plus, and NADP plus is converted into NADPH. So here we can say synthesis of both ATP and NADPH takes place. Whereas in cyclic photophosphorylation, only ATP was synthesized. Here synthesis of both ATP and NADPH takes place. Here again oxygen evolution takes place, means splitting of water takes place. Oxygen evolution is due to splitting of water, or you can say photolysis of water. This takes place at photosystem 2. Okay, the water splitting complex is present in photosystem 2. That's why in non-cyclic photophosphorylation, oxygen evolution is seen. Here the primary acceptor of electron is pheophytin. This is the point to remember. The primary acceptor of electron is, or you can say the first acceptor of electron is pheophytin. So this is the flowchart of non-cyclic photophosphorylation. Here both photosystem 2 and photosystem 1 are involved. Okay, photosystem 2 is activated by absorbing light of shorter wavelength around 680 nanometer, and photosystem 1 is activated by absorbing light of higher wavelength around 700 nanometer. So what happens here? Light is absorbed by the reaction by the pigments and present in the photosystem 2, and the reaction center gets, which is made up of chlorophyll A, gets excited. It releases electrons and the electron is accepted by the primary acceptor pheophytin. This is pheophytin. Okay, and then the electrons are transferred from one molecule to the other, and like I've already said, the electrons are transferred from the molecule having low redox potential or reduction potential to a molecule with high reduction potential. Okay, so here you can see primary receptor is pheophytin, which transfer the electron to plastoquinone. Then we have the cytochrome B6F complex. Okay, then we have plastocyanin, and plastocyanin finally transfers the electron to the photosystem 1. So here you can say the electron that were lost here is getting replaced by the electron which are coming from photosystem 2. Here you can see this also get excited, it releases electron, it loses an electron, and it is gaining the electron, or it is accepting the electron which are released from photosystem 2. Here, okay, and then again the primary acceptor here is FRS. And then the following and the final receptor of electrons are NADP+, which leads to the formation of NADPH. So here both ATP and NADPH are synthesized here, whereas in cyclic photophosphorylation only ATP was synthesized. And now you can also see that in photosystem 2, photolysis of water takes place. Okay, this is the water, splitting of water results in the formation of oxygen. So oxygen evolution takes place in non-cyclic photophosphorylation. And the site of occurrence of photolysis is photosystem 2. Okay, now let us discuss a little bit about photolysis of water. Now we already know that photolysis of water is nothing but splitting of water in the presence of light. Okay, and what are the products of splitting of water? First one is electron, then we have proton, and then oxygen evolution. Oxygen is also released. The water splitting complex is associated with the inner side of the thylakoid membrane of photosystem 2. So we can say the site of photolysis of water is photosystem 2. Some of the ions are involved during the photolysis of water. These are the major ions: manganese ion, chloride ion, and calcium ion. These three ions are involved in photolysis of water. And this is the chemical reaction of photolysis of water. Water give rise to protons, electrons, and oxygen. Now, what is the significance of photolysis of water? So first one is obviously the oxygen evolution. And some of the other importance of photolysis of water is that now we know it is necessary to replace the electrons that were lost from photosystem 2. Okay, so these the electron lost from photosystem 2 are replaced by the electrons that are coming from the photolysis of water. Okay, and the electron which are lost from photosystem 1. Okay, so these are replaced by the electron which are provided from the photosystem 2. Okay, so this is the significance of photolysis of water. Another important point to remember is that as the electrons are released from photolysis of water, these electrons enter the electron transport chain. Okay, and this transfer of the electron from one molecule to the another creates a proton gradient. And this proton gradient, which is created across the thylakoid membrane, is very important for the generation of the ATP. This we will study in the chemi-osmotic hypothesis later, how the ATP is generated by the breakdown of the proton gradient. First, the proton gradient is created by the movement of the electrons across the electron transport chain, and then there is a breakdown of this proton gradient which leads to the generation of ATP. Now let us study about the chemi-osmotic hypothesis. So chemi-osmotic hypothesis was proposed by Peter Michel in 1961. So what he proposed that ATP synthesis is linked to the development of a proton gradient across the thylakoid membrane. So here you can see this is the thylakoid membrane. This is the lumen of the thylakoid. Okay, or you can say inside of the thylakoid. This area is the stroma. Stroma is the fluid of the chloroplast. Okay, so what happens during chemiosmotic hypothesis? As the electron moves through the electron transport chain, protons are accumulated inside the lumen of the chloroplast. So from stroma, the protons enter into the lumen here. protons start accumulating in the lumen of the chloroplast, and there is high concentration of proton here. Here you can see there is accumulation of protons here. So this accumulation of protons creates a proton gradient, or we can say creates an electrochemical gradient. Now this electrochemical gradient is made up of two parts. First is electro and second is the chemical part. So electro means the charge of the protons. That's why we say electro, and we say chemical because there is a concentration of protons. Okay, that's why we say the chemical. So the electrochemical potential consists of the two parts. First one is the charge of the protons, second one is the concentration of protons. Okay, now what happens? There is a breakdown of this proton gradient. So what happens? There is a complex present here in the thylakoid membrane known as ATP synthase. So this complex is known as ATP synthase. This is an enzyme, also known as complex 5. This is also known as complex 5. I will tell you about this complex 5 later when we will study electron transport chain in detail. So what this ATP synthase is made up of two parts. Okay, first one is the FO part, second one is the F1 part. This FO part is embedded in the membrane and it is known as the transmembrane ion channel. It is known as the transmembrane ion channel. Whereas the F1 part protrudes out from the membrane. Okay, so this is the F0 part. Okay, and this is the F1 part of the ATP synthase. This F1 part, which is embedded inside the thylakoid membrane, this acts as a transmembrane ion channel, means it helps in the transport of the protons back to the stroma. This acts as a channel through which the protons get transported back to the stroma. And what happens during this transfer creates an energy, and there is a conformation change in the F1 part. Okay, and this conformation change in the F1 part leads to the synthesis of ATP. Okay, so simply you can say the breakdown of the proton gradient, which happens due to the moving back of the protons from the lumen to stroma through F0, which is present in the thylakoid membrane, generates ATP. So this is the end of the session, and we will be discussing later about the dark reaction and some of the other important points about photosynthesis. Thank you very much.